Welcome everybody to this introductory bioinformatics pre-webinar training. I'm going to be talking about what next-gen sequencing and bioinformatics are, first defining the terms pretty generally, and then talk about the applications of these technologies in public health, specifically looking at infectious disease programs across the U.S. All right, so again, the main goal is to make sure that everyone has this fundamental understanding of these technologies so that we can move forward next week during our training where we dive a bit deeply into all of these concepts that I'm going to be reviewing. So the first major goal of this is first defining what these terms mean, and then looking at how they're utilized to support investigations. And then we're also going to look at the three different phases of public health bioinformatics analysis so we can get some sort of conceptual framework around what we're going to be talking about through next week's training. So first and foremost, I think it's important for us to define what is bioinformatics. There's a chance some of you have started to hear these terms and think about these approaches, but I think it's important for us to first understand that bioinformatics really is this merging of disciplines of both biology and informatics, or data analytics, or computer science. And the way I generally like to describe it to folks is it's the application of computer science approaches to enable biological discovery. And that can be taken in a number of different ways. In general, you're going to hear bioinformatics being applied to the omics, or the different proteomic, genomic, and transcriptomic aspects of biology. So when we talk about DNA and we're using computer science techniques to analyze DNA, we're talking about genomics. In RNA in general, we're looking at transcriptomics. And when we're looking at the protein level, we're looking at proteomics. In public health and in regards to infectious disease surveillance, we're generally looking at DNA, so genomics, so applying bioinformatics to enable genomic discovery. And the technology that has allowed us to get here from DNA, the molecule, to data to analyze has been next generation sequencing technology. So we're using bioinformatics to analyze next generation sequencing technologies. And to give some context of this term next-gen sequencing, this is obviously the second iteration of sequencing technologies, the first of which being Sanger sequencing, some of you may be familiar with. And the main difference is that when we were looking at Sanger sequencing, we were looking at single DNA fragments sequenced in a single reaction. Whereas next-gen sequencing, the big defining factor is that it's a scalable biochemistry that allows for millions of fragments to be sequenced in a single reaction. So a term often associated with this is massively paralleled DNA sequencing. And the technologies to be aware of, especially in public health, is the Illumina MiSeq platform that invokes a next generation sequencing approach. Also available, more so you're seeing it in the academic realm, in some applications in public health, but the Ion Torrent PGM, and then also some folks here may be familiar of Roche 454. No longer a sequencing technology, but was pivotal to the implementation of next-gen sequencing. And so what we saw in the sequencing technology here is because of next-gen sequencing and because of being enabled to have massively paralleled reaction, you saw a drastic decrease in cost. So this is probably one of the more cited figures from the NIH, showing that around the implementation of next-gen sequencing, or at least wide adoption of these technologies around 2007, you saw a huge decrease in costs associated with sequencing. So with the decrease in costs became an increase, or led to an increase in accessibility. And so that's how you're seeing, at first in academic institutions, more and more applications and utilizations of next-gen sequencing technologies. And the predominant technology, or at least platform, being utilized for next-gen sequencing is one I pictured in that first slide. It's the Illumina sequencing platform. Specifically, at least what I've seen, is the MiSeq platform. And so the Illumina sequencing technology is also in line with most of the industry, is what's being most utilized in the public health space. Some laboratories may also have other iterations of Illumina sequencing technology, like the iSeq for smaller volumes, or the NextSeq for even larger volumes. But we're seeing mostly in public health are laboratories with access to the MiSeq platform. And this allows laboratories to sequence 16 to 30 bacterial whole genome sequences per run at around $200 to $300 per sample. This generates millions of reads, tens of millions of read data, each of which are around 150 to 300 nucleotides in length. And so just going over really briefly what's happening here. So when we're looking at analyzing next-gen sequencing reads, the data that we're analyzing is coming off of the Illumina MiSeq platform. And an important thing to note is that during the Illumina sequencing process, there's a step where we take the physical DNA molecule, excised from the organism, and it needs to be fragmented in order to be sequenced by the biotech or the biochemistry invoked in the Illumina sequencing platform. And there's a lot of nuanced steps in between how it gets from these short fragments to the read data itself. And I think there's a great video that's already generated by Illumina available on this YouTube link right here. But I think for this audience and to get an understanding of what we need to know so we can move on in the training next week, it's important to realize that the large DNA fragment, because of the limitations of the biochemistry of Illumina sequencing platforms, first needs to be fragmented into these 150 or 250, or I'm sorry, fragmented before sequencing can take place to generate 150 or 250 or 300 base pair read data. So this is what we call a short read DNA sequencing platform. So some of you maybe started to hear about long read DNA sequencing platforms like the MinION or the PacBio. Those are technologies available to the public health space. However, most laboratories right now are equipped. Actually, I take that back. I think all laboratories in the U.S., state laboratories in the U.S., are equipped with some sort of short read DNA sequencing platform. And then depending on your state, you may have also access to long read sequencing. And we'll get into the differences there next week. But I think important takeaway here is we're looking at short read DNA sequencing data, and that will lead into how we analyze the data to make some sort of public health inference. And so in public health, accessing next-gen sequencing technologies, we're seeing that have massive impact in both infectious disease as well as the newborn screening space. However, in respect to this training, we're going to be focused specifically on infectious disease surveillance and outbreak detection. So we're looking at things like foodborne, nosocomial, vaccine preventable, environmental, viral, infectious diseases. And then also, because we're looking at the DNA level, we also have that added capability of antimicrobial resistance screening, finding the gene presence or absence in a specific organism that would be an indicator of phenotypic resistance. And the implementation of next-gen sequencing in public health, I think most of you guys are already seeing this, this transition from pulse field gel electrophoresis into whole genome sequencing that was utilized or more so galvanized by the Genome Tracker and PulseNet programs by the FDA and CDC respectively. And so in this transition, these two organizations or programs help to put Illumina MiSeq platforms into every state public health lab in the U.S. And then alongside that came the bioinformatics capabilities to understand what this data means and how it will impact our lives as public health scientists. And then on top of that, the Office of Advanced Molecular Detection has also put a lot of funding and a lot of support into the implementation of NGS and bioinformatics technologies for public health labs. And this is what allows other laboratories to move even beyond just the PulseNet or genome tracker enteric pathogens of concerns, more so into the nosocomial or HAI, healthcare-associated infections, as well as environmental or vaccine-preventable diseases, like I listed in the previous slide. And the implementation of NGS at DCLS, I think, really mirrors well with that same trend, where in 2013, things started through the FDA's Genome Tracker Lab. In 2013, we had our first MiSeq instrument, and we sequenced 12 isolates that year, and we had a single dedicated ORISE fellow that was charged with leading that front. Over the years, we started to utilize more of the AMD funding, and this is also through the ELC grant and PulseNet programs. And we started to bring on more organisms and more sequencing capabilities. 2016 is actually when I joined DCLS, and we started to implement more bioinformatics capabilities, building our programs to make sure that we could analyze the data. We had scientists who were trained in understanding what that analysis meant. And we also had the compute infrastructure to carry out that sophisticated analysis. And as the years have gone by, we've increased our sequencing volume, and then also increased our organism spectrum. And now you can see just last year, we were sequencing CREs and group A strep, in addition to our PulseNet and genome tracker priorities. And of course, you've seen our team grow. Now in 2020, we have about 3 public health bioinformatics scientists at DCLS, 4 MiSeqs, and then around 4, I think maybe actually a bit more than that now, of wet lab scientists dedicated to generating next-gen sequencing technology for these, next-gen sequencing reads for these different programs. And the implementation of NGS around the region sort of also mirrors that same trend. All of our laboratories in the Mid-Atlantic region have MiSeq instruments. Most laboratories already have the capability to generate data for the PulseNet program, working to get that 8 of 8 in the coming months. And then about half of the laboratories in our region are now using this technology to sequence and support investigations for non-PulseNet organisms. And I will say too that jump from PulseNet organisms to non-PulseNet organisms, it's not so much the wet lab practices, because every single one of those programs is still using the Illumina MiSeq instrument, and a majority of which are also using the same wet lab protocols and sequencing kits and extraction kits and everything else included on the wet lab process. But the jump to get to non-PulseNet organisms is more so a challenge of integrating the bioinformatics approaches that are appropriate for those organisms. And those organisms, like I said, have included HAI, such as group A strep, or the carbapenem-resistant organisms, environmental pathogens such as Legionella, vaccine preventables, and viral sequencing as listed here. All right, and so I talked about next generation sequencing as a technology, and I also wanted to make sure that we had these definitions clarified in terms of next generation sequencing versus whole genome sequencing. Okay, so whole genome sequencing is a sequencing technique that works to sequence the entire genome of an organism. And in this case, we're talking about pure microbial isolates. So in this approach, all the genetic material from a single from a single isolate is excised and then sequenced using the next generation sequencing platform of your choice, mostly the MiSeq, like I had mentioned. And then that generates the sequencing data that is then analyzed using bioinformatics approaches to make some sort of public health decision. So in this approach, we have single isolates that are generating around 600 megabytes of read data using short read sequencing technologies. And just so, redundancy, because this is important here, next generation sequencing is the classification of technologies that allows for this massively parallel high throughput DNA sequencing. Whereas whole genome sequencing, WGS, is a classification of sequencing techniques in which we are sequencing the entirety of an organism's genome. And you could, in general, especially now with these technologies being available, we are generally utilizing NGS to carry out WGS. That is, we are using next generation sequencing platforms such as Illumina, the Illumina MiSeq platform, to carry out whole genome sequencing experiments. And that's the predominant technology we're seeing utilized in public health. The other experiments you might see NGS platforms being utilized for include amplicon sequencing, so targeted sequencing of very specific regions of a genome, or also metagenomic sequencing, where you're excising genomic content of an entire diverse population rather than a single pure microbial isolate. And so with whole genome sequencing, as carried out with a next generation sequencing platform in public health, that allows for bioinformatics to take place for two general categories of analytical techniques, where in one case we can use this short read sequencing data to infer genetic relatedness. And of course, there are obvious applications here that I'll get into in a bit, using sequencing data to infer genetic relatedness. But then there's also the category of analysis that I like to categorize as genome annotation. So seeing what genes are actually present within that short read sequencing technology. So first, looking into genetic relatedness, and I think this will have the most relevance and most easily comparable perspective in regards to pulse field gel electrophoresis. So during this transition, like I said, we went from PFGE. Oh, sorry, I see a little chat here. Okay, so we went from PFGE to whole genome sequencing. So I want to make sure that everyone has an understanding of what this transition means in terms of how it's impacting public health investigations of microbial or infectious disease pathogens. So when we were looking at the technology of pulse field gel electrophoresis, or PFGE, this was clustering bacterial isolates based on their genetic profiles. And this was always meant to be a proxy for epidemiological association. So the assumption here being that if isolates had a closely related genetic profile, then there is a higher likelihood that there's an epidemiological association between those isolates. So in that case, laboratorians can communicate that information to the epidemiologists, and the epidemiologists would have more resources, more data or more reporting to base their investigation in terms of where resources need to be allocated. And PFGE, of course, was the supporting technology for the PulseNet program for over 20 years. It was also utilized for other infectious disease surveillance, like I said, for HAIs, etc. And the way the genetic similarity was inferred was from banding patterns. So for those of you who aren't familiar with the technology, similar to whole genome sequencing, what you're looking at is pure isolates, and the DNA is extracted from those microbial isolates. And then it's allowed to interact with an enzyme that will degrade the DNA. And then the degradation can be visualized on a banding pattern. And the assumption here is that if the genetic content is similar between two isolates, it will degrade in reaction to that enzyme in a similar way, which will then be reflected upon the visualization of those DNA fragments. So we were looking at these genetic profiles and looking at their similarities inferred from these PFGE banding patterns. So when we're looking at next generation sequencing and utilizing that technology to carry out these whole genome sequencing experiments, we're still trying to answer that same question. We're still trying to cluster bacterial isolates based on genetic profiles as a proxy for epidemiological associations. The big difference here is that the genetic similarity isn't inferred by banding patterns, it's looking at the actual genomic information. So in this, we have the DNA molecule is fragmented, like I mentioned, and sequenced on these Illumina sequencing technologies. And we have all the data, the nucleotide data from those organisms. And using several different bioinformatics techniques, we can infer the genetic relatedness of these isolate sequence. So rather than inferring relatedness based on banding patterns, we're looking at these genetic profiles and comparing them from the actual genomic information itself. And hopefully you can appreciate the huge jump in data analytics that's happening here. Whereas before, when we were looking at banding patterns, we were distilling an entire microbial genome into 10 to 25 bands that we can visualize. Whereas when we're using whole genome sequencing data, we are sequencing the entirety of the genome using the millions of base pairs incorporated in the genome and comparing those computationally. And of course, this hopefully you can also see why this necessitates a computational analysis. No one is able to make these comparisons by hand. And so just wrapping that all up again. So these are both clustering approaches, where with PFGE, we're clustering by similarity in banding pattern, because a similar banding pattern means that the genomic content interacted with a certain enzyme in a similar way, which leads to the inference that they were genetically similar. Genetically similar organisms will have DNA that will fragment similarly in exposure with exposure to the same enzyme. And that's how the genetic inference was employed. Whereas with whole genome sequencing, we're starting to build these phylogenetic trees or allelic code differences, but it's still the same sort of approach. We're looking at genetic similarity instead of looking at how it reacts to an enzyme. We're looking at the actual nucleotide data itself and drawing these inferences. But nonetheless, it's still a clustering approach. Yeah, there was a great study in 2018. John Besser and the folks in the PulseNet program looking at Listeria, the utilization of whole genome sequencing for Listeria outbreaks over a number of years. And on essentially every metric measured, you can see that whole genome sequencing far outperforms pulse field gel electrophoresis in regards to the number of cave clusters that were detected, the clusters detected sooner by whole genome sequencing, and then the huge ones here too is the number of outbreaks that were actually solved in terms of identifying a food source associated with the clinical sources. One of the interesting metrics I think that a lot of epidemiologists are going to have to is going to change their roles in this pretty drastically, is also realizing that the clusters are now more closely defined, which is leading to smaller clusters. Sorry, I think I'm getting a little feedback from somebody. If you guys can all just mute your mics. Let me go ahead and try to do that on my end too. Okay. Yeah, so on every metric we found available, you're seeing whole genome sequencing technologies outperform pulse field gel electrophoresis. All right, and the two major methodologies you're seeing being utilized in bioinformatics to infer that genetic relatedness using the whole genome sequencing data is core and whole genome multi-locus sequence typing, or CWGMLST, or single nucleotide polymorphisms. And these are the two approaches we're going to go into depth with next week throughout the training. So just keep these in mind. I'm not going to get into the details of these today, but these are the predominant approaches for in terms of bioinformatics analysis being utilized for public health investigations. Oh, yep, and also, like I mentioned, the spectrum of sequencing capabilities at your laboratory. I think at your laboratory, everyone has access to Illumina MySeq, and the first priority seems to be in response to the funding and the programs from the CDC and FDA are these enteric pathogens, mostly carried by the PulseNet program. But then as your sequencing capabilities increase, it's really a product of what are your bioinformatics capabilities in your state or in your region in terms of what you have access to. And again, like I said, we were looking at the genomics level, and the added benefit there is that we can do more than just cluster detection from these, utilizing these data. We can also get into genome annotation. It's the same base biological molecule where we can make different sorts of inferences. And where we're seeing genome annotation utilized most is in addressing this global health phenomena, crises of increasing antimicrobial resistance around the world. And so in the same way, we're using a transition of technology from PFGE to whole genome sequencing for genetic inference. In the antimicrobial world, you're seeing a shift in technology from antimicrobial susceptibility testing to whole genome sequencing. With the big caveat, though, that there's definitely no intention for relieving AST as a technology, whereas PFGE has sort of been relieved of its time and duties in public health. AST is always an important aspect of antimicrobial surveillance. However, not every laboratory has the capabilities to carry out every AST assay that is required for surveillance. So, but like I said, every laboratory has the capabilities to generate sequencing data. So we can utilize all that sequencing data to help inform what's happening with the spread and distribution of antimicrobial resistance. And again, like I said, this is dependent on these next generation sequencing technologies and, of course, the bioinformatics capabilities to perform that sort of analysis. And the two national programs that are made incredible leaps on this front are the National Antimicrobial Resistance Monitoring System, or NARMS, and then also the Antibiotic Resistance Laboratory Network. Yeah, so wrapping all these things up, hopefully, you know, I've done a decent enough job for you guys to start conceptualizing that next-gen sequencing technology really is a revolutionary technology that is changing the face of public health. It's no longer reliant on banding patterns. What we need now is a whole discipline being implemented into the public health space that is bioinformatics. So utilizing these computational approaches to infer some sort of biological, or to make some sort of biological inference from this NGS data. And what we're seeing here is the biggest impact is changing how we're looking at outbreak cluster detection in terms of inferring genetic relatedness, and then also using these same technologies to enable for some really important use cases for genome annotation in terms of AMR screening, like I had mentioned. The other approach is also taxonomic identification, especially useful when we're looking at subspecies determinants such as serotyping for Salmonella, E. coli, or M typing for group A strep. And then also allowing us to look at what are the virulence factors that are present in a single outbreak, but also spreading in terms of what is emerging across the U.S. public health system. And so in terms of labs have now integrated these technologies with the sole purpose of supporting epi investigations. This is at least how I see it, and I don't think I'm alone in this, but it's the laboratory's job to support the epidemiological investigation. And that's the reason we're having this training with both laboratorians and epidemiologists, because you know, we realize that this is a different technology and it's a changing vocabulary and it's a whole different skill set, but we still have the same public health goals of identifying the outbreak clusters worthy of investigation that need intervention by our epidemiologists, but we're just taking a different sort of approach to getting to those answers. But because we are using a different approach, it requires, you know, a different language to be utilized between laboratorians and epidemiologists. We're no longer talking about PFGE patterns. We're talking about allele codes. We're talking about SNP differences, allelic differences, and all these different things. So regular communication, at least in Virginia and what I've seen in other states across the U.S. that have been really successful in implementing these technologies. The best practice is to continually communicate with your epidemiologist. And that's why we're at DCLS, we're trying to make that concerted effort of bringing EPIs and laboratorians to the same training so that both sides of the table understand what the technology is and also how to implement it and encourage the communication for each state there. And it's also, I try to always make a point, especially in these introductory trainings, to highlight that whole genome sequencing in bioinformatics is always meant to support or complement epidemiological data. It's never meant to supplant it or supersede it. This is always, all of the genetic inferences that we're making always needs to be contextualized with respect to the epidemiological data available to the EPIs. So this is always something to be a complementary conversation, not something that will be the single decision or the single data point that will influence the EPI decisions in that respect. So getting through the workflow of things. So we've talked about next generation sequencing technologies. I've mentioned that there are these bioinformatics approaches that allow for inferring genetic relatedness and also making these genome annotations. So that was really the goal of this quick pre-webinar. Next week, though, I'm going to be talking about bioinformatics. Like I mentioned it, and I've mentioned a couple of vocabulary terms in terms of MLST and SNP, but I'd imagine for a lot of the folks coming to this training, this is still a black box. Sequencing happens, and then bioinformatics says it's magic, and then we get these reports. Next week, I hope to draw back the curtains a little bit and educate everyone that's coming to this training on what are the bioinformatics techniques that we're employing that lead to our genetic inferences or genome annotation reports that we're generating. And when we're talking about bioinformatics, I like to think of it in these three phases that Lynch and the folks at Public Health Canada, I think actually British Columbia specifically, described in their primary and infectious disease bacterial genomics that I have linked down there below. And they talk about it in these three stages: the primary, secondary, and tertiary stages of bioinformatics analysis. The primary being looking at the data that was actually generated. And a lot of this is in regards to quality control. What does the data look like? Is it strong enough for us to make some sort of decision that will inform our public health action? The secondary analysis is doing some sort of data processing. So the raw data itself can't really be that helpful. We have to compare it to either another isolate's data or to some sort of database, or we have to do some sort of processing or manipulation of that data to make some sort of inference. And that tertiary part of analysis is actually making that interpretation, the biological interpretation based on the metrics generated from the data processing steps. So throughout the training, I'm going to be using this contextual framework to describe where we are in the analysis stages, so that it'll give you some sort of roadmap or context of what I'm talking about in terms of primary data analysis. We're looking at the quality of data being generated. The secondary analysis, when we're looking at how is the data being utilized, and then the tertiary analysis, like, okay, now that we've done something with the data, how do we make an interpretation of it? So that's the framework I'll be using. And again, I've got the link. I'll be able to pass the slides out to everyone after this training. So if you care to, feel free to take a read at this primary infectious disease bacterial genomics. But we will be getting into a lot of the same concepts that they covered in that paper there. So hopefully you guys got, walk away from this webinar understanding the definitions of bioinformatics and next-gen sequencing, and see how we're using these, both of these technologies to support public health investigations in both cluster detection, but also AMR or virulence gene surveillance. And then hopefully, I did a decent job of introducing to this cohort the three phases of public health bioinformatics analysis in terms of primary data, looking at the data generated; secondary, looking at how the data is being processed; and tertiary, looking at how the biological interpretation is being made. So that is it for our pre-webinar training. I'm going to go ahead and stop the share right now and then open up the floor to some questions. I'm sorry, stop the recording too.